here we have Sophia. Okay, and then IPSF Euro, where I am from, as well as Özge, who is in charge of the live stream, and as well as IPSF Emro and IPSF Afro, yes, who are unfortunately not here, but they also contributed to this event. So before this event, we also had a webinar, which you took part of, I presume, to get to know how to do Journal Club, and you will select it for the final round. So how this will go, you will have 7 minutes to present your case or present your article. After 7 minutes and 30 seconds, we will be deducting points. So make sure to stay in the time frame. And at 8 minutes, we will be muting you. So anything you say after 8 minutes will not be counted. So yeah, we will just follow the order submitted to you. And that's it. Good luck.
All right, so the presenter group 1 will be our team from my PSA Malaysia, TJC08. The group consists of, just 1 second. So we have Choon Fang, Alvina, Ya Lining, and Tan Khai Sin, which will be presenting over the topic of plant-based innovation in vaccine technology. All right, if you're ready, the floor is yours.
Okay, allow me to share my screen.
Yes, it should be available. Yeah, okay. Amazing. I'm sorry, just before you start, just a heads up. So I will be keeping track of time. When you start talking, I will start the timer, and I will put in chat 1 minute before and 30 seconds before the 7 minutes are up. So just so you're aware. So go ahead.
All right. Thank you, Philip. So I was just start now. So good day, judges and everyone. Thank you for joining us today. So we are from group TJC08. My name is Khai Sin. Together with me are Yen Fang and Alvina. So today we are going to explore a revolutionary approach to vaccine production, plant-based vaccines to combat infectious diseases, the current status and prospects. Next slide. So this slide shows the content outline of our presentation. Next slide. Before we dive into the innovation itself, let's understand why this research matters. So we understand infectious diseases are widespread globally with increasing speed and reach of human mobility due to many factors, such as the antibiotic resistance, climate changes, and wide travel of the virus through the air. Although there are already vaccines development, there is a demand of more effective, safer, and cost-effective vaccine to provide stronger therapeutic and preventive effects to the public. Next slide. And that is why here comes to the emergence of plant-based vaccines. So it was found that plant-based vaccine has the advantages compared to the conventional biologics. Among others, plant-based vaccines have the ability to grow in large quantities with lower cost, safer with a reduction of allergenic potential, and enhanced stability. We can see that several attempts of vaccines production were done with optimistic results. For example, more than 90% of the protection in chicken against Newcastle disease had been achieved in a clinical trial, while in another study, plant-based vaccine was developed against hepatitis B virus. Recently, plant-based vaccines get popular since COVID-19 pandemic. For example, the first plant-based vaccine against COVID had been successfully developed by a Canadian company with efficacy up to 70%. Next slide. So seeing the possibility of plant-based technology, this research aims to evaluate the current status of plant-based vaccines in combating infectious diseases. So for us to acquire a deeper understanding on its potential and future application in revolutionizing the healthcare. On top of that, we want to understand the limitations of plant-based technology in developing vaccines. So up next, I'll pass the floor to my teammates, Alvina.
Okay, thank you, Khai Sin. So I will present the importance of plant molecular farming vaccine. So what is the important? Firstly, it is an alternative strategy to the traditional vaccine. Second is the economic feasibility, and it can reduce the immunization costs. It also proved to be safe and effective. and rapid production and easily scale up, because each plant is bioreactor, and scale up is simply achieved by growing more plants. So because of stable plant molecular farming production, it can also withstand the temperature outside cold chain requirement. So this is an overview for the plant expression system. Next is the most of the study found out that the spike protein with the presence of the neutralizing antibody is the key target for the production of vaccine. So therefore, when we vaccinated, they can induce the immunity protection against the COVID-19 infections. Next. There are two methods. Firstly is the using the plant-based vaccine by transferring the subunit COVID-19 into the tobacco plant could be useful for the treating COVID-19 because it can provide the immunogenicity for the vaccine development. So plant can be exploited for the production of virus-like particles, which is a safer and can be produced in a large amount. So next, the second method is in this study is developing the specific antibody-mediated passive immunization. It provides a mediated protection against infection from highly contagious pathogen, and it can slow down the progression of the severe symptom. And additionally, adjuvant can be added as well to ensure sufficient immunity in the body can be produced. So next slide. So currently, this study is trying their best to develop plant-based vaccine for injection and oral immunization, and they are soon to be proceed to the preclinical stage, and next slide will be passed to my teammate.
All right. Thanks, Alvina. Moving on, despite we can see the potential of plant-based technology and the methodology, use of genetic material to stimulate the immune system rather than the conventional pathogens or their subunits is still in the early stages of development. So let's explore the main challenges in its development. Next slide. So the current technology involves identifying and incorporating a virus epitope into the plant host to be expressed as a vaccine. Ideally, the antigen should not get degraded because its breakdown can lead to inefficacy. So besides, the structural differences of complex glycans between humans and plants might affect the efficiency of a plant-based vaccine. For example, unconventional glycosylation may happen. This associated with the plant-derived antibodies may pose a potential immunological issue in the body. Therefore, it's worth the time for scientists to modify the proteins to be compatible with the selected plant for expression and scale-up process. Next slide. Up next, as the plant-based technology may employ transgenic plants with antigenic gene, purification of antigen from the plants, or simply take the edible parts with antigen. It sometimes causes lack of precision and accuracy of dose during the production and administration processes. If the intended dose is not administered according to the prescribed schedule, this may result in subtherapeutic dosing, overdosing, or dosing schedule deviation. Thus, strict quality control measures must be carried out to verify the potency and also the purity of a plant-based vaccine. Next slide. So last but not least, the scarcity of plant-based vaccine mainly attributes to the GMP compliance problems. We understand that various procedures and policy must be adhered to when manufacturing a pharmaceutical product. For example, the requirement of preclinical and clinical testing. As longer time and huge costs are needed, this situation greatly reduces the scaling potential of a plant-based vaccine. So next slide. So with this research, we hope for innovations that can fulfill appropriate manufacturing standards to ensure the smooth development of plant-based vaccines. With that, thank you for your attention and interest. Thank you.
Thank you very much for an interesting journal club. So the principle of making vaccines in plant is rather old, at least 30, maybe more years old, but still there are very few licensed vaccine. What is the main obstacle, in your opinion, to produce a vaccine in plants?
Oh, sorry, can you repeat the question?
Yeah, so the principle is rather kind of not old, but at least they have been trying this for at least 30, 40 years, but still there are very few vaccines that are produced in plants. What is your opinion about the main obstacles?
Okay, I think it is relevant to my part, the challenge part. So in my opinion, after reading the article, I think the GMP compliance problems may be the greatest attributes, because we understand that various procedures and policies must be adhered to when manufacturing a pharmaceutical product. However, when we're using the plant-based technology, using the open farming, it is not fulfill the requirement of the control environment. For example, you can see that the greenhouses may be required for transgenic crops to be grown. If we want to fulfill the GMP compliance for commercialization purpose, longer time and huge costs may be needed, and it is actually contradict with the benefits of a plant-based vaccine at the first place, because it reduces the scaling up potential and reduce the time-consuming benefits of this production platform, which is the plant. So I can say that lack of regulatory compliance is the main reason that limits its commercialization.
Thank you very much. Other questions?
Thank you, the teams. I think very interesting things. I think I agree with Tor that this technology is not new anymore. I mean, I encounter my previous schools also having vaccines growing from plants, but the problem is, I think, what we have mentioned. But I just want to get your opinions on what is your view on anti-vaxxer. You know, if this kind of plant-based vaccines will be going out, would there be a huge change in their opinions of you know, because they just don't like vaccines? Would it change your mind? What is your opinions on this?
In my opinion, this plant-based technology, this is the considered as a third generation vaccine. So it is completely, although we have these technologies thus far, I believe the development process is still in the we are still discovering the process. I think for example, to convince the industry or the company, manufacturing company, we have to have the quality control measures in place to verify the efficacy and the safety, because that is the two main things when we want to when we want to commercialize a pharmaceutical product. So in my opinion, I think having that documentation step is very important to convince the company and the industry.
Yeah, I think you get my question wrong. I'm asking about your opinions on, because there are a lot anti-vaxxer. So those people that anti the vaccinations, so I'm not sure that, like, would this kind of vaccines, if it come out from the markets, would it be a better choice or better acceptance among the anti-vaxxer? I'm not sure, you know, because probably they are worried about the source of the vaccines of the current technology. If this technology is coming in, so I mean, in the perspective of consumer, would that stand a better chance for acceptance? So I just want to get your opinions on this.
Yeah, may I add a point? So for anti-vaxxers, I think for sure that plant-based vaccine will have a greater acceptance among the users, because, for example, like the conventional biologic is using the pure animal part or the living pathogen. So with the plant-based technology, the product is expressed by the plants. So I think it maybe have the potential to convince the anti-vaxxers. So this is my opinion.
All right, thank you.
All right, thank you so much. That concludes the Q&A session for the first presenter, and we wish you good luck. now we will have the second group coming in. According to the order, the next group will be TJC. Let me just confirm. It will be TJC 02. Who will be presenting on the advanced dermal drug delivery system. So just want to call on Yasmin Ahmed, Puspa Das, and Hamza Hamri. If you guys are here, feel free to turn on your camera, and you could take this opportunity to start sharing. Okay. We have 2 minutes to give a prep. So Hamza, if you're here, could you hear us good? Oh yeah, 1 minute, please.
Yeah.
All right. Okay. Solve that. Thank you so much. Sorry to rushing. The second presenter is not here, so we have the delayed starting time a little bit. We have informed everyone at the beginning of the presentation, but then I realized that you weren't here. So sorry for that. Just to confirm that your team only have one presenter, or you would have two? All right, two presenter. Yasmin is going to be your co-presenter. Great. Whenever you are ready, feel free to share your screen.
Before I start, I would just like to say that I will be keeping track of time. As you know, you will have 7 minutes for your presentation. After 7 minutes and 30 seconds, we will be deducting points. And after 8 minutes, we will mute you, so nothing will be counted anymore. And I will send a disclaimer in chat when you have 1 minute left and when you have 30 seconds left, so you know where you're at. So whenever you're ready, go ahead.
Oh, wait. Sorry. I actually mix up the team order. It's actually supposed to be Sam, like the team from Nepal. Let me just follow up with them one more time. I'm so sorry, TJC02. Your presentation time is actually in an hour. Apologize for that. But however, let me follow up with Samadri, and if that's not the case, we also have our next presenter, who is Choon Lee from the UK. So I will just ask your prepare if you are prepared for the presentation. If yes, please give us a thumbs up, and we could give you the floor to give a presentation next.
He said he needs more time. It's no problem. We'll just contact the TJC 07 and see what is going on with them.
We could give it a couple more minutes. Isn't this like a student life? We are doing everything all at once, and now we have Other all the way from the UK practicing his fire drill and still give us a presentation during our competition. I believe that's a good sign. Okay, I believe that we could give the third team another three minutes. If they don't show, then we would proceed on with our next presenter. Okay, let's call it what's the word to say someone is like drawn? Like
Disqualified.
yeah, disqualified. Sorry. My English is not working really well right now. So in that case, other, just for your information, you have the privilege to start your presentation early if you could. If not, we could still wait another 5 minutes for you to prepare your presentation. Thank you so much.
As long as you're done with the fire drill, of course.
Yes, that's so unfortunate, but you know, he is getting the real fire to start this presentation, so we should really expect, you know. All right, I got a confirmation from the team from Nepal. They cannot participate and are now withdrawn from the competition. Okay, then the floor is going to be from Arthur. All right, so sorry that we
Right,
have to
sorry,
rush you up a little bit.
Yep. No, that's all right. Okay, so what do I do now? Should I share my screen? Should I?
You can
Yes,
take
you
a
should.
deep breath, enjoy
Okay.
your time, and
How does it
then
work?
get
You
ready.
can share your screen, and just I will give the same disclaimer as before. So you have 7 minutes. At 7:30, we will start deducting points, and at 8, we will mute you, so nothing will be counted after 8 minutes. And I will give a disclaimer in chat when you have 1 minute left and when you have 30 seconds left. So just so you know. Okay, but whenever you're
I've
ready,
got
you can
30
start.
seconds of grace period.
So 7 minutes.
No,
yes.
so like I got 7 minutes of presentation, then I got 30 minutes of
30 seconds.
buffer.
Yes.
Okay. Okay, it makes sense.
Excuse me, what's the number of this presentation?
This is
IJC1, I believe.
Okay.
yes.
Let me fully introduce you, officially introduce you then, Arthur. So next presenter, we have IJC1 from, give me 1 minute, 1 second. So his name is Choon from BPSA United Kingdom of Great Britain. He's part of the European Regional Office, and his presentation topic today will be the advanced dermal drug delivery systems. So as soon as you're ready, Arthur, the floor is yours.
Can I try if that worked on share link? Okay, that works. Okay. Yeah, I can start now. Okay, good afternoon, everyone from London. My name is Arthur. Today, I'm thrilled to talk about a very interesting paper on advanced dermal delivery systems. So today's paper will be about putting metformin in solid lipid nanoparticles in topical gel for dermal delivery purpose. Now, let's begin. So, I think I shouldn't be spending too much time on introducing metformin to all of you because I'm not surprised that everyone will know what metformin is, given it's one of the most common anti-diabetic drugs. But this is not what I'm going to talk about today. This study is actually focusing on a more novel way of metformin. So basically, it's to use it to prevent photoaging of the skin by sunlight, which disposes patients to skin cancer. And this UVB ray is the one that causing it. Now, you may ask me, Arthur, why do I show you a pharmacological diagram? Well, I just want everyone to focus on the circle here. So one of the ways that metformin works is actually inhibit the reactive oxygen species from NADH in macrophages, which coincidentally, that's how UVB induce. This is the pathway that UVB induce and cause skin damaging and skin cancer. So hereby, metformin serves as like a perfect countermeasures in preventing skin cancer here. Just one thing to note is that metformin is a very hydrophilic drug, making it a challenge to permeate the lipid bricks and mortar structure of the human skin. Well, they also identified that perhaps maybe we can change the chemical properties, but they also identified that this is a very complex task. So they end up coming with a formulation approach. And the formulation system they choose is solid lipid nanoparticles, which is a nanolipid emulsions where the drug is encapsulated in the nano-emulsion lipids. It is made by lipids, surfactants, and drugs. And here, the surfactants is a critical role because it helps to bind the aqueous and lipid phase together. And the reason why the research group chose this SLN system is because of better skin permeation, better stability, good tolerability, easier scale up, sustainable, and safety, and better safety outcomes. Right. However, as I mentioned before, metformin is a water-soluble drug. So this study is literally about talking about putting us water-soluble drugs in a pocket of lipids. Well, the author actually identified that no one has tried that before. And even if so, there are only a few studies, but none of them were about the effects of binary surfactants, which are the mix of hydrophilic and lipophilic surfactants with SON and API in there. So the objective of today's study is actually to evaluate the permeation effect of SON versus control, and to evaluate and characterize the properties of SLN metformin formulations from different composition, in this case, across different twin span ratio. Right, so these are just some basic and common materials for SLN. But the key here is that the ratio of twin 60 and span 60 here. And they use prop ultrasonic method to create the SLN. And they make 5 preparations, where the first one is a control because they didn't add a second surfactant, span 60 there. And for the second part of the discussion today, I'm going to talk about the permeation test they also performed here, where they use conventional metformin gel they make in laboratory as a control. Right. So this study talked about 6 elements, but because of time limits and the focus of today's study, I'm just going to focus on the first one, in particular, theta potential, which they use dynamic light scattering for the as a tool. Right. And for the second part of today's discussions, permeation test is an in vitro skin permeation test using rat skin. So they test the metformin level both in and through the skin using French cell systems and temperature at 32 degrees Celsius, close to normal skin temperature. So here's the results. But I'm going to simplify it so that we are not seeing this graph anymore. Okay, so as I mentioned before, theta potential is today's focus. But why? Why did the author chose theta potential? Because theta potential is actually a very important indicator for stability because of the charge of repulsion and steric stability that affect the coagulation risk and which could hamper drug release. What they found, which is something they expect in their literature review, is that binary surfactants actually give a more negative theta potential because of the dipole in the ethoxy group of the non-ionic surfactants, which gives a more condensed and packed coverage, giving a high density and higher charge. So this is expected. But what the author did not expect is that initially they hypothesized that smaller sizes of particles will give a higher theta potential because of higher surface area and higher surfactants and density. Now, I would like to draw your attention to the yellow and greens here, where you see F3 with the smallest nanoparticle size actually has a smaller theta potential than F4, which has a larger particle size. So what the research team then looked is the ratio of twin and span. And what they found is that actually the more close twin and span to 1-to-1 ratio, the more negative theta potential is. What this gives to us is that actually the amount of surfactant absorbed can change theta potential and potentially overcome the particle size challenge. So what is happening here, it gives some insights for future development. And I'm trying to press this and I can't press this. Okay, so the other thing is that about results and discussions that they find is that if they give too much span, actually this would defect the film layer and reduce the theta potential. And for permeation study, it's quite obvious that better permeation is observed and potentially giving better local effects. And there are 2 prospects here. One is to build that this study proves that SLN performs better. It builds in knowledge and confidence and use for drug company to use it in the future. And second is to help build a reference guide, especially on a twin span factor, so that future developer can better utilize it. One thing that I think this study could have done better is that if we look in the graph, that we see that actually SLM permeates way better than metformin. What we concern is that maybe it's too good that it becomes transdermal, and therefore further study have to investigate if that could be an option and find a balance in the formulations so that it can be optimized. Well, thank you for your time, and I look forward to the discussion in Q&A. Right. Did I make it on time?
So
Yes, you did. You did.
yeah, okay. Can I go? Can I go first?
Yeah, sure.
All right, thanks, authors. That's very fantastic presentations, clear and nice, and I think we grabbed a very clear ideas about the essence of the papers, and just some points I would like to get your opinions. Like, since that metformin is such a hydrophilic compound, what in your opinions that why this presents as a challenge for it to for it to permeate into the skins?
Sorry, are you asking like why metformin being hydrophilic and makes it difficult to permeate the skin?
Yeah.
So I mean, it comes with a very common principle about like dissolve like. So, I mean, the skin, as I said before, I can't find the slide, but the bricks and mortar structure, and it's basically a tight lipid packed together, and you can't just put water soluble substance in there easily, and that's why it can't penetrate easily.
Yeah.
Yeah.
Just want to make sure that you are clear with that, because I mean the skin layer, especially we are pointing out to the structural corneum itself, consists of the the major lipids is coming from your lipid bilayers, and also your hydrophilic parts is coming from your corneal sides with packed with the keratin. So you're actually having a sandwich layers of hydrophilic and hydrophobic or lipophilic properties. So you have to overcome these two things in order for it to go in. So we have a certain aspect we're looking at, which is called the log P. I'm not sure you touched that, but I mean that is the point. We also look at how easily for a compound. I mean, metformin definitely have a challenge because it's too hydrophilic. So, but that doesn't mean that it couldn't penetrate the skins. So another comment I would like to make is on your last point, where you're pointing out that this could be another benefits of being a transdermal drugs, and of course that will be also another thing. So we have to look back what I mean, any opinions that, do you think that even though it penetrates into this kind of amount, do you think that is this suitable for being a transdermal drug in this case?
Well, actually, from what I've read in this paper, this is actually based on a study that performed by someone else before, that they tried to put it as transdermal for diabetic patients. So in fact, I can say that it is possible because someone has done it, but whether that was optimized in other questions, because I think we also have to consider the patient factor in it, because so far the tablets were working fine, and given the conservative nature of the pharmaceutical industry, usually unless there's a huge problem with oral route, I don't think they would have an interest in delivering that as a patch or gel. But it is scientifically possible, but just not in the market because of various reasons behind it.
Yeah. You're right in somehow. I just have to make you clear is that for a transdermal drugs, we have to still fulfill that to make sure that the drug itself is potent. So with the little amount of drug being penetrated through the skin, and that is being immersed in such a big volume of the blood in our body, so you may not able to achieve therapeutic levels even. So that is what we might need to look into later. I mean it is a good point I mentioned, but I mean there are a lot of factors to be considered about the drug itself, and also the possibility for it to be a transdermal drug. Yeah. Right, that's it for me. Yeah.
All right, thank you so much. I would like to ask if Dr. Tor has any questions regarding the presentation.
Well, thank you for a nice presentation. I was just wondering, this is not exactly my field, but when you want to use liposomes for transdermals, is the size or the chemical composition of the liposomes most important, do you think?
Sorry?
What's the most important factor when you want to use liposomes in a transdermal drug? Is it the size or the chemical composition?
Well, I think both are important here. I mean, we're talking about formulation of pharmaceutics where, you know, different things suit different cases. And I think the idea, as this study showed, that actually the chemical composition, the twin span ratio, serves as a important and crucial indicator here, given different effects. So I would say chemical composition is important, but then we have to think about the target we want to get into. And in fact, the size of the liposome or the size of the carrier really depends on what barrier we are trying to get rid, and really depends on the target and the depth of the skin. So I think, as we talked about, the transdermal, maybe there will be a different, there's a completely different formulation for transdermal than dermal. And I can't just say which one is more important. I say it really depends on what are you treating and where are you treating, how are you treating it as well.
Yeah, thank you.
Fantastic. Thank you so much for the Q&A session, and good luck to your results, Arthur.
Thank you.
To inform you guys, Arthur is actually one of our junior participants, where he's a second year from the 4-year program. So this is actually very inspiring to see some students coming up and game their pharmacy school up to the limit and exceeding beyond. All right, thank you so much. You may now stop sharing screen and we will proceed with the next one. So I believe our next presenter in order is, if Philip could confirm the next presenter. I do have so many lists on my laptop.
TJC05. TJC05 should be the next one.
All right, then. Okay, TJC05, we have Fatima Ahmed Besrat and Fatima El Sahara Gouda. I hope I pronounced the name right. This is actually a group of a combination participant. So Fatima is from CPSC Yemen, and Fatima is actually from IPSF Egypt. They have been randomly assigned together and start working on this presentation, where it's going to be under the topic Advanced Dermal Drug Delivery System. So if we have Fatma here, I believe she was here, right?
Honestly, I don't know. Maybe we should contact them because I don't see them in the participants list now.
Oh, her name. I saw her name at the beginning, but I think she left.
Oh yeah. Let me quickly check again. All right. In that case, we could give them some minutes. I believe it could be a bit crashing with the schedules. Okay, she's replying. Just to confirm with her. If not, next in line would be a break, correct?
Yes.
Okay. Okay. Let me check again. So thank you so much for all of your patience. I'm still contacting them. I believe this is also a lesson learned for us organizer as well, that in the future we really need all of them here, even if the session is 2 hours long.
Michelle, are they replying?
Yes, they are replying. I'm giving them 2 minutes. So if they could join in 2 minutes. Somehow there was a confusion in time zone. Okay, so we are giving them 2 minutes starting from now. If they do not reply, we could give our judge a little break, and we could start with the other team in 15 minutes in that case. But let's just give them 1 more minute. Okay, somehow there would be one presenter, but then she's typing right now. So apologize for the delay. All right, in that case, we can call another team withdrawn. So then that give us a time to jump to the break right away. So thank you so much, judges, for your first half. We'll come back in let's say 8 minutes, because then we can start proceeding the presentation at 1:05 GMT plus 0. Okay, I'll take the pause on the recording, and then we can have a relaxing time.
And at 8 minutes, we will just mute you, so nothing said after that will be counted. I will give you a heads up in chat when you have 1 minute left and when you have 30 seconds left. Don't be scared, you still have enough time. That's just like a heads up for you to know where you're at with the time. And whenever you're ready, you can start, and I will start the timer.
Okay, maybe I will start now. Ladies and gentlemen, have you ever heard about plant-based vaccine? If you haven't heard about it, I, Erica, with my teammates, Samuel and Nafisa, are going to discuss about COVID-19 plant-based vaccine that are currently in phase 3 clinical trial. In 2020, the COVID-19 pandemic became a global problem that resulted in many deaths. Until now, the impact of COVID-19 is still a big problem in several countries. This has resulted in a large demand for the COVID-19 vaccine. However, improper vaccine storage and serious adverse events are a significant concern, with a lot of vaccine need to be distributed with liquid nitrogen and so on, making the distribution of vaccines under optimal conditions to communities in low to medium economic countries difficult. And this has the potential to disrupt the stability of the vaccine, thereby affecting its efficacy. This problem led to the discovery of CoVVAX, the first COVID-19 plant-based vaccine. It uses the virus genetic code that are delivered to the leaf cell of Nicotiana betamina using a harmless bacteria that assembled to virus-like particles, as intended to be a co-VLP vaccine candidate. Co-VLP is stable at refrigerator temperatures, which make it easy to use in remote communities and in low and middle income countries. CoVVAX works by helping your body to protect itself against COVID-19 by producing both antibodies and immune system that recognize the virus. The early phase provides data regarding the safety and immunogenicity of the vaccine, with the phase 1 study being a dose-finding study in healthy adults on a small amount of subjects, advancing to 3 subgroups according to age and comorbidities in the phase 2 study. However, the clinical efficacy have not yet been determined in those studies, and there's a clinical question to be solved. Does the positive result of the phase 1 and 2 trial translate well clinically? And yes, this is the answer. This journal club discussed the efficacy and safety of CoVVAX in the phase 3 clinical trial. The phase 3 clinical trial of CoVVAX is a double-blind randomized controlled trial with the plan shown. And the important thing we should note from this table is the outcome. The outcome of this phase 3 clinical trial is the absence of symptomatic COVID-19 cases identified at least 7 days after the second dose. 25,000 volunteers were assessed for eligibility, 1,000 participants were excluded, resulted in 24,000 volunteers that were randomized in a 1-to-1 ratio between the active vaccine and placebo groups. Subjects were given 2 doses spaced 21 days apart, and the efficacy data were collected at least.
Right, so the efficacy of this vaccine was analyzed in both the intention to treat and per protocol population. The primary efficacy measure was the absence of symptomatic COVID-19 cases identified at least 7 days after the second dose, with the vaccine efficacy shown on the slide. The safety data of CoVVAX comprised of solicited and unsolicited adverse events, presented here in percentage. Of particular note, no life-threatening safety concerns were observed, and most were transient grade 1 or 2 events. Overall, this trial has shown that CoVVAX can effectively prevent symptomatic COVID-19 at least 7 days after the second dose, and the adverse events were mostly mild to moderate and transient. Specifically, 99.4% of the safety population had less than grade 3 adverse events. The local and systemic adverse events that were observed include local injection site pain, headache, myalgia, fatigue, and malaise. Moving on to the internal validity of the study, the investigators included both the intention to treat and per protocol population in the analysis, and the efficacy results are quite similar. The intention to treat result accounts for all of the future patient populations that may not receive the full scheduled doses of the vaccine, while the per protocol results account for those who received the doses as intended. The similarity between the two results indicate that the treatment has a consistent effect between the ideal scenario and in the potential real-world scenario, further enhancing confidence on its efficacy. The incidence rate of COVID-19 occurrence was given in person-year denominators to account for the progressive imbalance between the two groups. This trial was also conducted multinationally, which enables the efficacy to be measured on different race and ethnic groups. However, the median follow-up period of 2 months was quite short for the long-term efficacy to be measured. And based on the trial results, the efficacy in individuals aged 65 years and over and in severe COVID-19 disease cannot be determined due to the limited number of subjects in these subgroups. And so we might not be able to recommend this vaccine in these populations.
The first study showed efficacy of vaccine virus platform against the Delta variant of the COVID-19 virus, which at the time of the publication is dominant strain around the world. Here we can see that the vaccine demonstrated a comparable efficacy among the other vaccines. But it should be noted that although this vaccine has been approved in Canada, it has not been approved in other countries due to limitation in production. Standardization of claims used for defense production is more difficult than standardization of other vaccines. In conclusion, COVID vaccines are adequate to prevent COVID-19 in adults aged 18 to 65. Before we close our presentation, we will say vaccines play a huge role in the development of plant-based vaccine technologies. Promoting further research into plant-based vaccines for various diseases can revolutionize medicine by offering sustainable, effective, and accessible solutions for global health challenges. And that's all about our presentation. Thank you.
Yeah, thank you for a very nice presentation. So my first question then is, what was the main advantage by producing this antigen in plants as opposed to, for instance, in mammalian cell cultures? What are the main advantages of plant production here compared to a cell line, for instance? Did you hear my question?
Well, thank you for the question. I think the advantage is that a plant-based vaccine is less dangerous. I mean, they uses plant cells to create harmless version of the virus called virus-like particles, and these are non-infectious and unable to replicate, meaning they can mimic the virus without the risk of mutation like DNA vaccine. And even though mRNA vaccine that has lot in COVID vaccine doesn't have mutation risk, they are read by ribosome to replicate into antigen protein, especially mRNA vaccine that is self-amplifying type. And the ones like AstraZeneca use this technology and has a risk of overexpression, thus triggering unnecessary immune response. Also, plant-based vaccine is very stable. You can store it in refrigerator up to 30 days and it will stable in room temperature up to 6 hours. And the other vaccine with another platform, I think it's not very stable, and the distribution handling is quite difficult than the plant-based vaccine.
Yeah, thank you.
All right. Thanks to the team for the presentations. So I think following the question just now, I just wonder that from what there have been trials here. So looking at the potential of these vaccines, what is your opinions on I mean the challenges that are coming with the plant-based vaccines? And I mean, as for now, you're aware that it's only available in Canada and it's very limited elsewhere.
Okay, so I think I'd like to answer your question. So we have identified several factors that maybe challenges that we may face in the development of plant-based vaccines. So the first being the plants for the vaccines are growth in variable conditions. So it might be difficult to standardize the growth conditions for the optimal production of the antigen. And so the second one, we believe that plant variability is also a major challenge for the development of plant-based vaccine, and also it may be difficult to scale up from the small-scale laboratory production to the large-scale commercial production to maintain consistency from the quality of the plants.
All right, thank you. I think I have no further questions as for now.
Thank you so much for the Thank you for the presentation. Now I would like to call for the next group to present, the TJC14. Are you here, guys?
Okay. Let me check TJC14.
Yes, I think Shoruk Shaban, sorry for the pronunciation, is part of that team.
Yeah, that's correct. All right. I would like to fully introduce them to our presentation. This is also a mixed group of Raut Hotul Faisun from TPAYC Turkey, from Euro office. And then we also have Charuk Shaban Oma from IPSF Egypt and Shad Ayub from IPSF Egypt. So this group will give a presentation on the topic of advanced dermal drug delivery system. All right, team, whenever you're ready, please start sharing your screen and the floor is yours.
So Shoruk, this is your time. If you are here, please unmute yourself, start sharing.
I think Rofa will be the one who is giving presentation. Rofa, yes, we cannot hear your voice. Could you try one more time? I'm so sorry, your voice is very inaudible. If there's a suggestion that you might be able to change your microphone input, or if you do have headphones.
Okay, right now, can you hear my voice?
Yes, that's
perfect.
the best. Do you have a slide presentation?
Oh, yeah. All right, so I will share my screen
Amazing.
presentation.
Before
But I'm
you,
so sorry, I think I have a problem with the camera, so perhaps I
it's
will
totally fine. You can start sharing, and while you do that, I will explain the timing. So you will have 7 minutes for your presentation. At 7 minutes and 30 seconds, we will start deducting points. So if you go over that, you will be losing points. And if you pass the 8-minute mark, we will mute you.
Okay.
Can you hear me?
Okay, please wait a second. Yeah, I can hear you clearly. But
Okay, so if you pass the 8-minute mark, we will just mute you, and I will give a heads up at 1 minute left and at 30 seconds left in the chat, so keep an eye out for that. And yes, we can see your screen now.
okay, perhaps you can see my screen right now.
Yes.
So I guess.
Yes. Oh, wait.
Yep. Okay, I know how to make it.
Just press slideshow, I think. It's loading. Just a second. No?
Can you see my screen?
We just see the screen of the presentation. So not the full screen of the slideshow, just the editing part. Maybe you can go into Zoom and change your, so stop sharing and then start sharing again without exiting the presentation, if you know what I mean.
Sharing. Okay. I'm so sorry. Shuruk, are you here?
She's in the Zoom. I'm not sure if she is here.
Yeah, I'm here. I'm just
Okay.
I try to make it on my device.
Yep. Can you please share the screen? I think I have the problem with the screen right now.
If it's a problem, you can also send us the link and we can assist.
Of course. Yeah.
Sharing. Yeah. Just drop the
Of course,
link
I will.
in the chat and then I can
Yep.
start sharing for you.
Of course. Thank you so much. Sharing. Okay. Right.
Okay. So whenever you're ready, you can start. I will start the timer.
Okay, thank you so much. So hello everyone. So my name is Rafa and my teammates here is Shuruk. Today we are going to present our journal discussions. The research paper we are bringing to you today is about the formulation and characterizations of curcumin nanoparticles for skin cancer treatment. So why do we think that this paper is important? So we found that the paper, this paper is contributes by helping us understanding the curcumin's nanoparticles characterizations, such as poor solubility and rapid degradations, and how its nanoformulations can address these issues. So this studies is also helping us understanding skin cancer treatment using nanoparticles. So in these slides, we can also know the advantage using the dermal therapy systems. So the drug delivery through the skin could enable the better treatment through the site-specific administration. It could also decrease the side effects because if we use the oral delivery system, it may cause so much side effects such as first pass metabolisms, and it could also hurt the other organs probably. And then the skin dermal therapy systems can also have the high bioavailability. So next. Yep. Shuruk.
Okay, our method is the study design. The study design is a lab-based experimental study. Our inclusion criteria is experimental subject, 8 weeks old, abdominal shaped skin mice. The exclusion was not clear in this study. The material used is our active substance, the curcumin powder, the polyvinyl alcohol, it was a surfactant, and the grade E100, this is a copolymer. This is the thing we loaded the curcumin on, and the HPMC gel that used for stability purposes. The intervention and the comparator, the intervention is a curcumin nanoparticle loaded at HPMC gel. The comparator is just a placebo plain blank. And the procedure, they prepared more than 20 formulations. They used at least five formulation for each parameter. The study parameter is the concentration of the polyvinyl alcohol, injection time, steering rate. The optimal was 650 RPM, and the ratio of organic to aqueous phase. The optimal pH is 4.5. as a primary and secondary endpoint. Our primary endpoint is the size and the polydisparity index, zeta potential of the nanoparticles, has a loading capacity, encapsulation efficacy, and the in vitro drug release at pH 4.5 and 7.4, and the skin papilloma development and survival rates in mice. The other secondary endpoint, the morphological characteristics, the statistical comparison of in vitro drug release of different pH. The statistical analysis, they just utilize the t-test performed in MS Excel to determine the significant difference between the two pH's value, 4.5 and 7.4. The results was that the p-value was less than 0.05, considered to be that there is a significant difference if there is any change to the pH. Next, the results now.
Could you please? Okay. Now I will present the results. Next, we can go to the next. So the results here, we have the PVA concentrations surfactant results. So the importance of the PVH surfactant here, it is really important to targeting the molecules, the cancer drug molecules into the cancer cells. So it can be seen from the results in figure 1 here that there is a very low and higher concentration of PVH has a prominent effects of the physical stability and the particle size of the nanoparticles. So the size of particles in low concentration of PVA, it tends to settle down over time. And then the higher concentration of PVA is also leads to the formulation of the microparticle, which is something that we don't want. So it is considered, so I'm sorry, can we go back to the previous? Yeah. So it is considered that the PVA concentration in the middle here is the perfect concentrations. And then also there is the figure number 2 in the right side. It is actually similar with the figure number 1. So with the increasing volume of the aqueous phase here, the particle size of the nanoparticles is also increased. So on the basis of this data, the volume of 4 milliliter nanoparticle was selected as an optimum aqueous phase volume. Next. All right. We can go straight to the discussions. Yep. Next slides. Okay, in the discussions, we are going to discuss about the critical findings, the study comparisons, and the critical appraisal of this study. Next. So the discussions, we found that the objective of the study is to formulate the nanoparticles for the skin cancer treatment. And then we found the critical findings that it is really important for the pH of the E100 that was polymer, because it has the acidic characterizations of the molecule, so it will enhance to the cancer cells, and it will use for the targeting therapy. Next. Also, we found that the role of the stability formulation here is also important, which is PVA. PVS is surfactant, and then also the slow injection, because it will determine the I think we have out of time.
You can finish what you have to say in 10 seconds, and then
Okay,
That's
Shuruk,
it.
you can continue.
I can say fast. The appropriateness of this study is the objective to develop a nanoparticle so this can address the current challenges in current cancer therapy. They use control and in vivo testing. Characterization also of nanoparticle make an importance. Next.
Unfortunately, that is out of time, so we are past the 8-minute mark now.
Let's go straight to our Q&A session. So, judges, the floor is yours to ask questions.
All right. So thanks the teams for the presentations again. So a little bit disturbing. I think there's some background noise, but anyway, so my questions coming here is, from what you have come across from this paper is that why nanoparticle would be the first, I mean, would be the preferred choice over here to use to target a skin cancer through through the skin.
Yeah.
Okay.
Yeah. Sure.
Okay.
Please.
Okay, for the nanoparticle have a very, very small size, so it can permeate the skin very fast. And it also the reason that they use this type of nanoparticles that the cancer cells are of acidic pH. So this acidity of the pH, the optimum one was 4.5, as the research said. So it allow more permeation and more efficacy than the conventional ones.
Yeah. So
And also,
yeah, just sorry to interrupt a bit. I mean, you're aware that this is a polymeric type of nanoparticles, which is a solid type, but not if I'm correct, because I don't have the full papers. From what you have explained, that it's a polymeric nanoparticles, right?
Yeah, it's a polymeric because they use the Edo grade, I think E100. This is a copolymer they use, and they use polyvinyl alcohol as a surfactant, so it aids the solubility, and they use it for loading, and they use also the HPMC as a hydrogel. This is for stability purposes because it's resistant to the temperature and resistant to humidity, and it make a good film on the skin.
Yeah, so you may get confused with the concept of nanoparticles, because I think this is a solid type of nanoparticles that it doesn't get through the skin as easily as the liquid type. So liquid type of nanoparticles will be like your emulsions type, nano emulsions or micro emulsions, or even your liposome. So those are the liquid type of nanoparticles. So I'm looking at here, this could be the solid types. So for the solid type of nanoparticles, the penetration isn't that good as compared to the others. So you have a big challenge here. So and as far as I concerned, solid type of nanoparticles doesn't penetrate the skin. So most of the time it will just accumulate at hair follicles, for example, your epidermis of the skin.
Yeah.
So
Sorry, can I sorry to disturb you, but yeah, we have acquired we have acknowledged that the solid nanoparticle is really low in the permeability and then low in the solubility I have mentioned before. So the combination we are bringing the new formulation for this nanoparticle, which is the curcumin, and also we are using the HPMC and the other one, the PVH, the surfactant that I was mentioned before. The combination of this one, these molecules make the more hydrophilic agent and reaction. So it will combine. It will detect. It will attach to the water, and it will make the solubility really higher for the release of the drug itself. So the new combination of this nanoparticle can make this nanoparticle, this solid polymer nanoparticle, into become more solubility. Because actually we have mentioned in our slide, but we don't have time for the results. So yeah, we have the graphic for.
Thank you for your answer. I'm so sorry, Dr. Ko. Maybe we could pass on to Dr. Tor for his question, and then we have to conclude the Q&A session for this team.
Yeah, I know we are running out of time, but I have just one question. Since you ran out of time with the conclusion, what was the conclusion? Did the formulation have any effect on the treatment? Did they see any effect on the tumors?
The effect of they tested it on the rats, and it showed that the development or the growth of tumor were decreased when they applied the curcumin the polyvinyl alcohol gel on the skin of the rats. So but all of this is just preclinical. So the limitation of the study is that it need to be done on clinically on real humans to test the effectiveness of it on humans.
Yes. Thank you.
Thank you for your answer, and that was a wonderful presentation and Q&A session. I would like to call in our next presenter. Next, we have our speaker, individual participants. His name is Usama Muhammad Anwar Muhammad, IJC 10. Mr. Muhammad will be presenting over the topic of, I believe, just one second. Mr. Muhammad, if you have time, please feel free to start sharing your screen. So he will be also presenting on the advanced thermal drug delivery system. Just to remind the judges, he's also part of the our best junior participant. So he's second year from the PharmD program. So Mr. Muhammad, as soon as you're ready, the floor is yours.
But first, just let me give my disclaimer out for timing. So you have 7 minutes to present your presentation. At 7:30, we will start deducting points, and at 8 minutes, we will just mute you, so everything you say will not be counted. I will give like a reminder in chat when you have 1 minute left and when you have 30 seconds left. So now you may start, and I will start the timer whenever you're ready.
So, hi everyone. I'm Osama Muhammad. I'm a PharmD student, actually going Arising level 3. So, for the scenario that I was working on, I chose a review paper that's talking about the latest advances in hydrogels and TDDS systems. So first, like the agenda for today is we're going to go through the 5 sections of the paper: the introduction, the challenges for TDS, and hydrogels as drug carriers, and what kind of vectors are used in hydrogel systems. And then at last we're going to be going through the conclusions. First, the paper talks about the drug delivery systems as in general, and talks about the importance of TDS in modern medicine and the development of effective medicine. Talks about then they go through the structure of skin and things related to how the drug works. Next, they dive deep into the challenges for the transdermal drug delivery systems. They talk that the skin is an efficient barrier, and that the main challenge is for the hydrogels to go through the stratum corneum. What else? Okay. Okay. And they talk about differentiating between the dermal, transdermal, topical, and they're differentiating how the drugs work on each and every skin layer, either the epidermis, dermis, or hypodermis. Next, they talk about the hydrogels as drug carriers, which is basically the main part for this, because they're trying to talk about the latest advances in hydrogels as drug delivery systems. They mentioned that hydrogels are 3D matrix, which allows it to hold in more water than other types of gels. They mention other types of gels such as bigels, which are a mix of hydrogels and organogels. They talk about mu gels, which is like an emulsion and the hydrogel. They're comparing these types, and they're talking about how the crystalline structure of mu gels hold it from working efficiently with hydrophilic drugs. Then okay. Okay, then they describe the difference in structures between the matrices of all the different types of gels that I mentioned before. Fourth part, they talk about the vectors and hydrogel systems. They talk about the nanocarriers, that they can be different types, such as physicals, emulsions, droplets. They talk about how the nanoparticles or the nanocapsules can be loaded with drugs that can be incorporated with the drug particles themselves. Okay. And yeah, that's it for the vectors and the gel systems. In conclusion, they talk about how hydrogels have a great area of improvement and how it can be used for delivering different types of drugs, but mainly hydrophilic drugs, because they're trying to find other ways to make it work with other types of drugs, but so far nothing. And I think that's it for the presentation. Okay, can you guys hear me?
Yep.
Hello?
Yeah, I think I may just go first for the questions. So thanks, Usama, for your presentations. So I'm just curious about, I think this should be a review paper that you are trying to, trying to review on the website. So it will have a lot of things to cover. I mean, just to get clear in your mind is that why potentially hydrogel is a good platform for dermal or transdermal drug delivery system. And in fact, I mean, there have been lots of gel formulations even for transdermal drug delivery. So what would it be? I mean, a good platform, because I think I wasn't clear from your presentations to this particular point.
Okay, hydrogels in general can uphold a huge amount of water due to its 3D hydrophilic structure or matrix, and they can absorb the drugs and they can encapsulate it, and then they're one of the best platforms for latent or prolonged release of drugs in transdermal systems.
All right. Okay. So I mean, of course, there are more than what you have just mentioned, and I think one more point that I would like to highlight is that you are trying to say that the hydrogels would work only for hydrophilic drugs, but not for hydrophobic drugs.
They said it the highest efficiency is with hydrophilic drugs, and they said they're trying to make there was, as I read, they said that they have been some not inconveniences but irregularities with hydrophobic drugs. But it works best. The hydrogels work best with hydrophilic. Organogels works best with both. Biogels also with both, and imugels work best with hydrophobic.
Yeah. So I think that is the things that I would like you to pinpoint, because I mean, hydrogel is just a very basic platform where people started to add more things into it. Then they call it different names. That, I mean, of course, because it's a hydrophilic system, so you have a challenge to incorporate hydrophobic drug. That's why you're coming in to incorporate the emulsion that probably you can carry a hydrophobic drugs into it. Yeah, I think that's it for me. I think I'll give time for Tor to ask some questions.
Yeah, so you chose to present a review paper, and that's of course okay. It doesn't fit exactly into our scoring formula, but after reading this article, what was kind of the main take-home message for yourself? What did you learn from this article that you didn't know before and that you think is important for you?
Basically speaking, I thought that hydrogels or hydrogels in specific are just a dead end. But as I read through these papers and the reference papers that were presented at the end of the paper, there is a lot more area of improvement that I thought there would be. But overall, the review paper is just presenting the latest advances in hydrogels as a drug system delivery. Drug delivery system, sorry.
Okay, thank you.
All right, I believe that marks the end of the presentation for our third to last presenter. Next, finally, we have the actual presenters from TJC. 1 second. C2. Yes. We have Yasmin Ahmed from LPSA Lebanon, Puspa from FOP Nepal, and Hamza Hamri from Algeria. So this group is also having junior participants, and they will be presenting over the topic of advanced dermal drug delivery systems. So team, whenever you're ready, I know you have been ready for 1 hour, but just for your say again, whenever you're ready, please feel free to share your screen, and I wish you both and the team good luck for your presentation.
You already heard my disclaimer, I believe, so I will not be repeating it, but yeah, 7 minutes, 7:30 directing points, 8 minutes we mute, and I will give reminders, and good luck.
So can you all see my screen?
Yes.
Okay, so Yasmin, you can go ahead.
Hello, I'm Yasmin Begdash, and I'm going to be presenting my Team Journal Club 2, Hamza Hamri and Puspa. Today, we are here to discuss an important research paper entitled Bioavailability Study of a Transdermal Patch Formulation of rivastigmine compared with Exidin in healthy subjects. And it was published in the European Journal of Drug Metabolism and Pharmacokinetics, which had an impact factor of 1.9 back in 2022. Our journey begins with a critical problem statement. We are here to explore whether an advanced dermal delivery system can overcome the skin barrier and provide a controlled release of a therapeutic agent. Alzheimer's disease, a progressive neurological condition affecting millions worldwide. The chosen treatment, rivastigmine, is a cholinesterase inhibitor, but how does it work? Rivastigmine operates by inhibiting the enzyme acetylcholinesterase, responsible for breaking acetylcholine in the brain. This inhibition leads to increased acetylcholine levels, crucial for cognitive functions. Now, let's explore the transdermal drug delivery system, a convenient route of administration, especially vital for Alzheimer's patients. The system ensures controlled and sustained drug release. The transdermal drug delivery system functions through a specialized patch adhering to the skin surface. This patch contains the medication, which is slowly released and absorbed through the skin, ensuring a steady therapeutic effect over time. The objectives outlined in the research paper were to first evaluate the bioavailability and bioequivalence of multiple patch applications. Second, to assess safety, efficacy, patch adhesion, and skin irritation, prioritizing patient well-being. As we embark on this enlightening discussion, we are about to uncover a piece of research that could reshape Alzheimer's treatment. Prepare to witness the power of this paper as Hamza Hamdi from Algeria takes the stage to discuss it.
So now moving on the method they used in this bioavailability study. So they used a randomized controlled phase 1 study that was conducted following the good clinical practices, obtained the necessary regulatory and ethical approval, as well as the volunteers' consent. So it doesn't pose any ethical concern. It was conducted during 2 5-day study in which consisted of the investigational patches took place, and it was conducted on 31 healthy male adults who successfully completed the trial. When it comes to the inclusion criteria, so it was well documented in the research paper. They included healthy male adults aged between 15 and 50 years old, with a body mass index ranging between 18.5 and 30 kilograms in order to cover various body types. None of them were smokers, and again, the informed consent was already obtained prior to the interventions. When it comes to the exclusion criteria, it was mainly chosen to ensure that none of the subjects and the risks for the administration were included, and it was also well documented in the study paper. For the interventions, they used a RIV TDS test formulation developed by Liui Pharma, and an already available and commercialized drug, Exelon, developed by Novartis. For the results, initially there has been 50 screened subjects, 36 were randomly assigned to the treatment, of which 34 received the medication and only 31 completed the trial. It was shown that the test formulation had a better skin adherence as well as dermal response, and it has both drug has systemic tolerability in accordance with the safety profile of the drug substance. Now, the strengths of this study, it demonstrates that both formulations are bioequivalent. It, as an advantage, it also assessed secondary elements like skin adherence and tolerability, not only the bioequivalence aspect of the drugs, and of course, the pharmacokinetics, safety, patch adherence, and skin irritations, all of them were evaluated. As for the weaknesses, we see that the small sample size of 31 subjects, as well as the short duration of the study, they pose a weakness for this study. As for the inclusion, both drugs were included bioequivalent. The test formulation showed an improved pharmacokinetic profile as well as skin adherence. The study involved an already marketed drug, the Exelon, which is a transdermal drug, which has a transdermal drug delivery system with a controlled release pattern, which provides an undeniable evidence that pursuing this drug delivery route is efficient and possible for the researchers, which answers the case of our journal club competition. And also another advantage is considering the controlled release pattern over 24 hours. It helps with increased adherence with the Alzheimer patients, as well as it reduces the workload on the carers like the nurses, since there is only one application per day and there is no need to check on the patients like twice a day if it was an oral medication, for example. Now, moving on to the literature evaluation, we see that the study had an appropriate design with the randomized assignment. It had valid results with 90% confidence intervals, and however, it was only performed in a short duration of time. Whereas comparing it with other studies, we see that there is a scarcity of studies that are evaluating the bioequivalence of such a drug, which poses which indicates that this is still a novel area of research where further research is still needed, and the improvements of such a drug delivery system is still an area of the future to be explored. And that's a wrap. Thank you for your attention.
Tor, do you want to go first this time?
No, you can go first. That's okay.
All right. So all right. Thanks, Yasmin and Hamza, for your presentation. So I think this is a little bit different from the rest of the topics we have today. So this is a clinical trial based journal clubs. So I mean, you pinpoint some of the things that is needed as for clinical trials, but I'm just curious about, I mean, the current products that you are trying to bring out as compared to the innovators. So what are the advantages, or what are new, you know, what are advanced over there as compared to the innovator products?
Yes, so according to this study, it was more of a generic under development by a new pharmaceutical company, and for their product, they shown that it had a better pharmacokinetic profile in comparison with the already available marketed drug by Novartis, and also it had gain comparison with the other drug. So not only it was bioequivalent, it was also more. It had performance than the already available drug, which can be a competitive advantage for them, or a marketing sales point when they bring it to the market.
Okay, so from what you have identified the shortcomings of the current trial, so what is your opinions on how to improve the current study if you were given a chance to redesign the whole trials? Do you have any ideas?
So yeah, I think one of the shortcomings of the study was the short number of volunteers. But I think this is also an area to explore by including maybe the Alzheimer's patients, who were excluded from the study since it was only a phase 1 bioequivalent study. The other thing, depending on the outcomes of the research, like the areas for improvements in terms of skin adherence, in terms of safety, in terms of irritation, those are the parameters that I see are good to, are some areas of improvement where improving the performance of the formulation can make a difference and should be investigated further.
All right, thank you.
Yeah, I have a question related to that, because you said that the problem with the study is few participants, but that's kind of dependent also on the effect size. If the effect size is great, then maybe you need fewer participants. What was actually the effect size in this study? Did you mention that during your presentation? I didn't get that. Well, how much better was the one patch compared to the other?
Yes, so one of the parameters was the area under curve. So the new formulation showed a bioavailability of around 84%, whereas the commercialized drug was around 74%. So that's a 10% improvement in bioavailability, which was shown in the research paper. Yes, we didn't the chance to include this in the presentation, but it was already documented.
Okay, so with the number of participants they had, they were kind of able to show a statistically significant change in effect.
Exactly, yes.
Okay, thank you very much.
Thank you.
All right, that was a fantastic presentation. And with a group that comes from different region, I believe that was a challenge that you have to work, not just coming up with the idea, but also, working together in a various time zone as well. All right. Last but not least is come to the final, and we have one last presenter imported all the way from California. Good morning. And I would like to introduce all of you to our last presenter of the day. So his name is Mr. Joseph Miles Asurin. He's from APHA ASP United States of America and a member of Pan America Regional Office. Mr. Joseph today will give in the presentation over the topic of advanced dermal drug delivery systems. So Joseph, whenever you're ready, you can start sharing screen now, and Philip here will be reminding you about the time, so I'm just gonna give you a heads up that he will send you a message saying that it's running out of time. You have 7 minutes, and if you go over and exceeding 8 minutes, we are going to mute your microphone and jump to the Q&A session right away. So in that case, I bid you good luck and have fun as the last presenter of our journal club competition.
Thank you. Also, I wanted to say thank you also for accommodating me, given the time difference. Quickly, I just wanted to ask, can you see my screen? Yes, okay, perfect.
Yes.
All right, so again, my name is Joseph. I'm from the APHA ASP, United States, and I'll be presenting on the study clinical efficacy of tropical tranexamic acid with microneedling and melasma by CAR in 2020. And it's just to explore the role of TA, or tranexamic acid, in enhancing advanced transdermal drug delivery, or ATDD. So just to give some context for microneedling, it's a form of ATDD, and it's where we have microneedles creating microchannels that helps assist the drug delivery inside through the skin. This is less invasive and painless compared to other treatments such as like hypodermic needles. And there are different versions of microneedling shown below. And then for melasma, it's a reoccurring condition or disorder of hyperpigmentation that causes like darkening in the skin and uneven coloration that is hypothesized to be caused by biosynthesis of melanocytes. And these can cause adverse effects in the quality of life for said patients with histology features including solar elastosis, basement membrane disruption, and increases in vascularization and mast cells. For current treatments, one of them being microneedles, they are unsatisfactory and unpredictable. For tranexamic acid, currently it's being used for bleeding conditions and disorders such as uterine bleeding, but it has actually recently been used for skin brightening as a topical treatment. And then how it kind of works is that it's actually a derivative of L-lysine, which helps it be more acceptable in the body. It actually has like 4 different mechanisms that can be used for skin treatment, including inhibition of the melanin synthesis, reduction in the plasminogen pathway. It's an anti-inflammatory and anti-angiogenic. And just to kind of go in a little more depth of how it kind of works, you can see in the middle, we have the plasminogen activator that actually gets blocked by TA in its connection with keratinocytes, plasmin, and melanocytes, which can all lead to helpment with the hyperpigmentation of melasma. For the study itself, the objective was to assess the clinical efficacy of topical TA, 10% with microneedling in melasma. The study size was 40 subjects with the inclusion and exclusion shown below. Inclusion being that there needs to be a diagnosis of melasma and the patients need to be between the ages of 18 to 50. And then the exclusion there. As for the clinical evaluation that was done prior to the testing, all subjects were like classified in terms of how their melasma was and what type of melasma they had. And then in terms of severity, these were all measured with modified melasma area and severity index, or MMASI, for the entire face. Interventions included the test group, which of course is the 0.5 to 1 ml of the 10% TA, as well as the control group, which is the distilled water. And then just to kind of show, the study design was a split phase, prospective randomized open label study. So all 40 subjects were microneedled throughout both sides of the face, and then the left and the right were randomly chosen between 10% TA and distilled water as the control. This would be the initial treatment at week 0, and then treatments were repeated at weeks 2, 4, 6. And then on the 8th week, it was a follow-up. And at each time there was an assessment of clinical images, MMSA scoring, satisfaction, and side effects were recorded. For the results, you can see here, the TA test group had a 65.92% statistically significant improvement based on the MMASI scoring, with 72.5% of the subjects reporting excellent to good response, as opposed to in the control group where it was only 20.75% improvement and 72.5% reported partial to no response with the control group treatment. As well as for the safety and the adverse effects, there were no notable differences in the different side effects for both sides, both groups. For key takeaways, the study supports the use of TA in enhancing the use of microneedling. It actually mentions in the paper that they believe that in the control, the 20% improvement was associated with the use of microneedling. And this could show that its potential in enhancing other forms of advanced transdermal drug delivery systems. Tranexamic acid itself has many mechanisms that can help manage melasma and other skin conditions and disorders, and has much potential in that field. But of course, further studies are needed. Other microneedles and other physical advanced transdermal drug delivery systems. Limitations for the said study is that since it was open label design, there's poor blinding. Potentially, subjects could know which side is which in terms of the treatment versus control groups. Limited sample size, again, being 40, it's difficult to generalize this findings to larger populations. And since it was isolated in the same area, it could be a source of bias. As for the short follow-up period of just 8 weeks, longer follow-up periods are needed just to see how much the improvement has persisted in the treatment, as well as the microneedling process itself wasn't really well documented within the paper. Only one form is shown, and more studies are needed with different designs and tests prior to making this a common use therapy and treatment option for patients with melasma. And these are my references. Are there any questions?
Yeah, I have a question. Maybe you said it, but what were the downsides of using this drug? I mean, what were the side effects on these patients? Did you say that?
Yeah, so these were the main 5 that were shown from the paper itself. Most of them being more associated probably with the kind of the needles itself. Specifically, they mentioned they highlighted the dryness, but they said it's because of those microchannels actually allowing intradermal fluid from being released in the skin itself, which is why both sides had the same experience.
But if a sizable chunk of the control group has these kind of side effects, do you think this is acceptable side effects? In your mind, is this a...
Yeah, I was kind of looking through it. Most of the topical agents that we'll actually use are oil-based for people to avoid dryness. In this study, both control and the treatment were water-based ointments, or I should say creams. So in this case, it'd be probably ideal to modify it in a way that allows maybe some type of common moisturizer, which was not used in this case.
Yeah, thank you.
All right, Joseph. I think this is something interesting because I think tranexamic acid is having a huge debate. I think in any part of the world. So I think it's off-label used right now, still for the skin whitening, the so-called skin whitening. So
yes.
Yeah. So I mean, like yeah, I'm not sure what's the future for this, but I think potentially there has been some clinical evidence showing the potential of it to be used as a treatment for melasma patients. So in particular, this paper is looking at the combinations of microneedles with tranexamic acid. I mean, like, what do you think that the ideas having here? I mean, instead of just having the tranexamic acid formulation itself, I mean, if that would work the same, so yeah, what is your opinion on this? Like looking at the initiative of having these studies.
Right. When I was looking at different object, or I should say substances and medications that could potentially enhance ATDD, what I found interesting about TA was that since it had several mechanisms of action that actually work along the skin, some common things being that since it's anti-angiogenic and anti-inflammatory, things associated with, like, let's say redding in the face, or like different issues pertaining to like skinning in general, like for like in this case hyperpigmentation, but I can think of it in terms of like actually skin stretching. One of these being like solar elastasis, which is kind of like the UV caused wrinkling in the skin. I feel like cosmetically speaking, there's a lot of potential for TA and allowing for tighter skin, less inflammation throughout, less redding, because of its different mechanism of actions. And because there were no like differential side effects, at least in this case, it seems to be a much safer alternative than maybe more invasive or oral therapies that can affect throughout the system of the body.
Yeah, I mean, like, I think the ideas of having a microneedle here, that is the reason for it to have a shorter periods of investigations over here, because it will get straight away the drugs to go, I mean, into the dermis layers for its actions. So I don't see a longer period of time that would benefit. I mean, if this approach has been taken. So yeah, I mean, there's still a lot of clinical trials ongoing for this particular compound. So yeah, I mean that's it for me. Thank you.
Thank you. Yeah. If I could just add on, I mean, it could be also because the microneedling also did just speed up the whole process. But again, like, it's one of the limitations for the study. It needs to be done for a longer period of time.
Yeah, I mean, like, microneedle has been used for cosmetically for scar reducing, you know, so
Yes.
in a way actually helping also to regenerating the collagens to help in perfecting the skin and bring back the kind of texture that they want at the same time by improving the skin tone and colors. Yeah. All right. Thank you.
Thank you.
Okay, thank you so much, everyone. Thank you so much to all the participants. You really did amazing. Every single one of you had amazing presentations, and congratulations to coming to the final round. And a huge thank you to the judges as well for your amazing insight. We are now moving into the calculation period. I believe Manshari is already calculating. So bear with us as we calculate, and we will announce the winners shortly. Until then, would someone like to share something? Maybe the judges who would like to comment on all the participants, maybe give them some compliments or maybe something they can improve on.
Yeah, I would like, I share your opinion that I think these presentations were very good. They have obviously read their articles very carefully, and they have all kind of being able to really present for us the main conclusions of the papers that they have read, and it's also impressive that they can do this sitting in different cities and share these insights with us. So I'm very impressed with what I've seen. Thank you to all of you.
Yeah, I think the same for me. I mean, it's a good opportunity, I think, for us to get you know, to keep abreast with the latest things ongoing, even though it's not from my field, especially for the vaccine side, which, I mean, it's something interesting for me to learn again. At the same time, I think it's also a good thing for every I think a pharmacist to be over here, that to critically look into published work and learn to appreciate the findings, and also to critically look into the limitations that will help them in future to design a better research work if they are still continuing in that, or in practice. I mean, you have to look up for the best evidence ever to really helping your patients or physician to make the decisions. I mean, that is the point that I think that hopefully this kind of events or exercise will help. Yeah.
I agree.
Yes, absolutely. Especially because pharmacy is such an ever-evolving topic. We really do need to keep track of all the new medicine, all the new technologies that are coming up. So this was, I hope, very educational for all the participants. But yeah, maybe some participants would like to share something until we wait for Mashari's calculations. If not, then you can maybe grab yourself a drink and we will shortly. Or are you done, Mashari?
I am done. Oh,
We're
so
done.
excited.
Okay, no time for
Come on.
drinks.
When it comes to calculation, you have me. So I'm actually done. I believe this is going to shock everyone because so let me just confirm one thing, because the number is really, really amazing. And just for your own information, I just want to confirm with everyone here that we do have three teams with the junior participant in the team, including let's say IJC 10. And then we also have TJC 02, who is also having one participant who is the second year of 6 years. And then I believe IJC 1 is also still junior pharmacist from his school. So in that case, drum rolls, guys. If you don't mind, I would like to make this a very beautiful presentation. So bear with me. I am preparing the announcement, a proper announcement. So just one second. I know everybody is probably excited. I'm excited as well. Could you keep me company, Philip? Just talk to me right now.
Okay. What am I doing?
We can just talk about something while I'm preparing this presentation and announcement. How do you feel? Wait, we could talk about this just for your information. How has it been hosting this competition? Let's talk about this. It's been a challenge, right?
Well, yeah, it's been a challenge because, as you guys know or may not know, this was a collaboration between all 5 regions of IPSF, so European, Pan American, African, Asia Pacific, as well as European Mediterranean. So 5 regions, 5 regional projects officers, 5 different work styles, 5 different time zones, which was the biggest issue. So yeah, I mean, it definitely was a challenge. We had a bunch of ideas. We started this back even before summer. This event, this started off as an idea and then it evolved into this. But yeah, now we are doing this journal club. We're also doing a literature review competition in parallel, which might have been a bit too ambitious on our parts, but we are here, we are doing it. And yeah, this is actually the last day of our mandate, so that's exciting as well.
Yes.
But yeah,
I mean, it's been a challenge to host, apparently not just to us, but also not just to participants, but also to us organizers, because IPSF have 5 different regions, and then we all have different time zone and also commitments, and it's been a pleasure to host this competition. I just want to say, personally, I've been enjoying every single minute of it. Still it's hard because we involve too many parties, and also we understand that if it was also difficult for you in terms of communicating with your teammates, and we have some group as well that have been assigned, and their, let's say, their ability didn't match up in a way, because it was a challenge in terms of communication, language barrier, and also time difference. And I would like to thank you all here so much for staying up until today, come to the presentation on time, and still giving your best ability in presenting the your work. And journal club isn't an easy thing to do. So yeah, I mean, I hope this event bring you guys to the place where you belong. if you don't make it to the to the event, or let's say if you're you do not get a prize today, it doesn't mean that you didn't do your best. It was just that somebody might have enough time to prepare, and maybe they chose the topic that is very aligning with their work. So just from me, I know that everybody have been seeing my email at all time, at every minute of the day, and you have been getting answer, not getting answer. But I just want to say that we did our best in accommodating every one of you, and we are so grateful to have participants. So I'm about to share this screen, but let me just quickly confirm. Okay, Philip, do you want some music? Like when it comes to announcing the winner?
Did I play the music, or what do I mean?
I mean, maybe we might need a drum roll. How would we be able to do that?
Let me see.
Oh God, so sorry guys. I like to make things. We like to make things big. All right, the poster presentation is ready though. So
maybe do you hear if I do this?
Okay. Yes.
Okay, I'll just be the drum roll then.
Yes. Let's just make a drum roll, like how do I say this? Okay, how about we have Philip, could you screenshot the announcement, and we will allow prize winner to say something, and as a final remark. So which one should I start first? The best junior pharmacist award, or which one? Philip, could you help me choose?
Maybe, yeah, best junior. We could go for best junior.
Yes, okay. So just for you all information, we come up with this prize just because it's not an easy task for anyone to start doing research work, because it's been a challenging to like everyone, not just yourself. I believe all judges of all of our judges are still dealing with research daily, and it's a a topic, and it's a work where we evolve. Should we use the word evolve? Let's say learn every day. We have to keep ourselves posted and inform ourselves that we are in this and still working on that. All right, so in that case, let's start it with our Junior Pharmacist Award. This prize goes to drum roll. Congratulations to Usama Muhammad Anwar Muhammad from IPSF Egypt. You have made it to the excellent contribution by junior pharmacy students. So once again, congratulations. And would you like to say something? Is he still here?
I believe, unfortunately, not.
Unfortunately not. Okay, that's all right. Okay, all good. I believe we can send him an email later. And now let's move on to our actual prize. Let's go from our second runner-up. The second runner-up is, oh God. Congratulations to Mr. Joseph Miles Asurin, all the way from California, APHA ASP, United States of America. So the floor is yours in case you want to say something. Yay!
Thank you for the floor. I just wanted to say thank you for accommodating me. I know it's early on my end. I know it's late for yours. But again, I really appreciate all the effort that the team has put in. This was definitely fun and eye-opening. It's something that I kind of want to look into maybe further for potential research. It's funny, one of my friends, when I was kind of showing him my idea for the research, was asking if I wanted to go into cosmetics. I don't know yet, probably not, but we'll see. It's very interesting in terms of the field of pharmacy, how things can change and everything's always evolving. So thank you.
All right, thank you so much. You know, we already have our judge here who's a very specialized. So one of the key finding that I mentioned is always utilize our contact, because Dr. Go is actually, you know, his research is actually in the, you know, in this area, and I believe he's also have the expertise. And, you know, you never know, maybe one day you'll travel to Malaysia and the collaboration starts here, right? Okay, I think Usama is back. Hi, Usama.
Yes.
Have you heard the news?
What? And I know actually. I just there has been an issue with my internet.
Okay, in that case, everyone please pretend that you didn't hear anything. So we will go back to announcing the best an award, the first award. So congratulations, Usama Muhammad Anwar Muhammad from IPSF Egypt. You actually have been awarded the excellent contribution by junior pharmacy students. So we are actually giving the floor to everyone who get the prize. So I would like to ask if you would want to say something.
Oh, well, thank you. I seriously thank you so much. And aside from the fact that I really expect that I would have done a better job, I just really got very confused and just stressed out. But I thank you so much for real. Thank you so much. And I'm so happy about this. Like you can't imagine. So I really don't know what to say. I'm just speechless at this moment. Seriously speechless.
Thank you so much. I really hope that this award is going to you and it will become a start of something. I believe that, you know, in our profession, we are trained to be pharmacists, but research will also explore so many more opportunity. And thank you once again for participating, even if you are, you know, still second years. All right. In that case, let's just change back to our announcement. It comes to the first runner-up. Okay, Philip, can I have a drum roll?
Horrible.
It's okay.
I don't know if you can hear it.
No, we can't. So it's all good. All right.
I'm just jamming my table for nothing. I'm sorry.
Okay, let's
pretend
not
you hear
make,
the drum roll.
let's not make the participant wait no longer. Congratulations to TJC02. Yasmin Amira Bektash, LPSA Lebanon, Puspa Das, FOP Nepal, and Hamza Hamri from Algeria. You have been awarded the first runner-up of the Interregional JC Competition 2023. So the floor is yours. Feel free to share your thoughts.
Well, it's really one of the great surprises, I'd say. It was a very nice competition, learning wise and network wise, and I really thank you for organizing such a competition, for the professors, for being the judges. I mean, it was a bit challenging at first with organizing ourselves, especially given the different time zones and the different engagements. And I'm really grateful for the opportunity and for the prize. And I want to thank my teammates Yasmin and Puspa for doing great. And yeah, thank you very much. And I pass it on to Yasmin if she has something to say.
Yes, thank you so much. I'm very happy that we won. And I really thank my team, Hamza and Puspa. We were really collaborating very well with each other. And literally, it was one of the best teams I've ever worked with. And I'm so grateful. Thank you so much.
Okay, I was taking pictures, doing an organizer job. All right, okay. In that case, here comes the first, wait, not first runner-up. Here comes the winner of this IPSF. Wait, it should be the first ever though, because this is the first time we hosted together. The first ever IPSF Interregional Research Collaboration 2023 Journal Club competitions. So, drum rolls again. Last prize today goes to IJC1, Choon Lee from BPSA, United Kingdom of Great Britain and Northern Ireland winner. Yes, Arthur is also the other name that he goes by. So if you are here, please
Yeah, I'm here.
feel
Are
free
you for
to
real?
share your thoughts. Yes.
Are you for real?
Yeah, for real.
I mean, I am speechless because I ran out of time, and then I didn't know what I was doing a week ago, and then oh wow, that's amazing. I mean, I've never done journal club before. This was exciting, even though you have to read the paper for like hours and hours. But still, it was fun to learn about the topic. Especially, I may say that pharmaceutics is not my strength in pharmacy course, and that's why I was like, I have to keep on cross checking in my if my concept and theory in this surfactant thing was correct or not, and oh wow, I'm speechless, honestly.
You've earned it, and
Yeah, thank you.
you know, as a
Yeah, I
junior
mean, thank
pharmacy
you for
yourself.
organizing this event.
Yes.
Yeah, I mean, thank you for organizing the event. It was very fun for this summer, and then I mean, moving on, yeah, I look forward to other events similar to this.
All right. Okay. Thank you so much. Now I believe we have come to the conclusion of finally the IPSF Interregional Research Collaboration. For those of you who also submit your literature review, I would like to say that unfortunately we still haven't been able to collect the all the evaluation from our judges. So we'll do our best and we'll announce it hopefully by the 3rd of October. But we all will not be here. So our incoming RPOs from each region, or just from the organizer, will send you an email announcing the winners in that case. So once again, congratulations to you all. I would love to take this opportunity to take pictures with you guys so that we can record this moment of the history. And as a representative of all organizers, I would totally love to send a regards to Dr. Tor and also Dr. Go for accepting this invitation. It couldn't have been better without you guys. And I would love to say that I would probably not win anything because of your questions is so, insight, and your experience are so amazing. So I hope our paths cross sometimes. And if you guys haven't had a chance to check our judge profile release, Dr. Tord is actually from University of Oslo, Norway. And he was the one who proposed the topic of plant-based vaccine. So if anyone would like to check his research and any particular things that you would like to be in touch with him, please feel free to do so. Both judges are more than welcome to support you guys. And Dr. Goh here is from Asia Pacific region. He's also part of the IPSF alumni. So he has been in touch with us since he was a student. And now, once again, he come back to be our judge. So who knows? Each of you in this room might become the judge one day or might become one of the sponsors to our future students. So, please do not keep your hopes dry. Please keep practicing in research because it will always be the challenge. And I mean, I don't have anything else to say. Philip and Sophia, do you have anything to add on?
Just a huge thank you to the judges, the participants, and obviously all the RPOs and everyone who worked on this event. Everyone did such an amazing job, and I'm really proud of everything.
Yeah, basically just would like to say thank you for all of you to spend time to be here with us on a Saturday. So thank you so much.
Okay, all right. Then it comes to the time where we are going to take pictures now. Let me just, if you could help me, Philip, we could also take pictures together as well. So, all right, we still have some participants here who hasn't turned on the camera. If you could feel free to turn on the camera now. We're gonna start taking pictures, and we will send the certificate of participation to everyone, and this recording will be posted on IPSF Euro. So you will be able to rewatch your presentation, and maybe if you haven't been awarded this time, you could take that opportunity to, rewind of what happens and learn from the part that you can improve upon. All right, okay. Now, all together, let's take a picture. Okay. 1, 2, 3. All right, one more time. 1, 2, 3. All right. Okay. Thank you all so much once again. Please feel free to leave the room now, and I bid you farewell for the last day of our mandate. Thank you, judges. Thank you, Dr. Ko. Thank you, Dr. Tor. I wish you have a pleasant rest of Saturday, and we will be in touch with you.
Sure. Goodbye.
Bye.
Thank you, everyone. Goodbye. Have a nice day.
Bye.